Hey everyone, we're going to go ahead and get started. So I want to welcome everybody to our webinar on detecting shared mis-splicing across blood cancers using long-read single-cell RNA sequencing, and we're excited that you decided to join us this morning. So to introduce myself, I'm Alex Sockel, and I'm the segment lead for cancer genomics at PacBio. So I'll be giving a short introduction today. And I'm honored to be joined by Dr. Nathan Salamonis, who's an associate professor in the Division of Biomedical Informatics at Cincinnati Children's Hospital, to share some of his work on using long-read single-cell RNA sequencing to detect shared mis-splicing across hematological cancers. Just a few housekeeping items. First, just due to the number of people calling in, we've muted the attendees. Both talks you'll be hearing today are pre-recorded, but Nathan and I, as you can see, are both here live, and so we'll be joining the live Q&A session after the recordings. And we'll also be making the recording of this webinar available to you in the next week or so, so be on the lookout for a follow-up email with a link to the recording. The two presentations we have for you today are going to be followed by a live question and answer session. So you are welcome to submit your questions during the talks by typing them into that area provided on your attendee control panel. So up there with the question mark, and then you can enter a question. And then we've also uploaded a few pieces of complementary literature to your control panel on that little icon with a paperclip. So please feel free to download those and take them with you. And then lastly, there will be a brief 2-minute survey at the end of the webinar. So please do take the 2 minutes to fill it out, as it helps us understand your needs and plan for future webinars. So with that, I will start the intro. Hi, everyone. Thanks for coming to the webinar. I'm excited to tell you today about how accurate long-read sequencing from PacBio is enabling the next generation of cancer genomics by providing comprehensive and multi-omic characterization of somatic variants within the cancer genome, epigenome, and transcriptome. So to start, I'll first explain how our long-read sequencing works and what we mean by a hi-fi read. So these are reads that are both long, up to 25 kb, and also accurate, so greater than 99.9% accuracy. You start with a high-quality, high molecular weight DNA molecule, and then you ligate on these hairpin adapters, which we call smart bells, to create a circular molecule. A single molecule is then loaded into a tiny well on the smart cell called a ZMW, where it binds to a primer and a polymerase, and the polymerase then reads around and around the circle multiple times. And because our errors are completely random, we can then use those multiple passes around the circle to create a consensus read. And it's this consensus read that we refer to as a hi-fi read. Another really cool thing about hi-fi is that we're actually able to directly detect modifications like methylation for free with no additional library prep needed. And we do this by looking at the kinetics of the polymerase as it's incorporating each fluorescent nucleotide. And so what's useful about this is that it takes the polymerase longer to add a base if the preceding base is methylated. It basically works like a speed bump for the polymerase. So we can actually read out the polymerase kinetics and use that to infer methylation status of both 5-MC and 6-MA. So what you get off the sequencer is A, C, T, and G, obviously, but also 5-MC and 6-MA. What hi-fi enables for cancer genomics is the ability to do comprehensive and multi-omic profiling of all of the somatic changes that are occurring in cancer. So this is across the genome, the epigenome, and the transcriptome. And what we mean by this on the DNA side over there in green, it lets you go beyond looking at small variants like SNVs and indels to also look at large structural variants, to be able to phase those mutations and then reconstruct the tumor's evolution, and then to overlay methylation status directly onto this information to understand changes in gene regulation through the epigenome. In addition to that, we also have the ability to look at chromatin accessibility using a method called FiberSeq, which uses a methyltransferase to label regions of open chromatin, which can then be read out by the sequencer. And lastly, for RNA in pink, which is where the majority of this webinar will be focused, hi-fi lets you go beyond gene expression to look at things like differential isoform usage, discover novel fusions and expressed mutations, and we'll hear a great deal more about this from Nathan later in the presentation. So before we get to that, I'll show examples for each of these where HiFi has been used to identify new biomarkers and power discoveries about cancer biology. So here's an example of whole genome tumor normal sequencing of a melanoma cell line. And I really like this example because it shows structural variant calling, SNV calling, phasing, and methylation all in a single plot. So to orient you here, we're looking at the P10 locus, and you can see the two normal haplotypes on the top there in brown and purple. And then in gray below, you see the data coming from the tumor. And so in this plot, we can notice a few things. So the first is that all of the reads coming from the tumor appear to be derived from normal haplotype 2, and we can tell this by looking at the pattern of SNPs present in the tumor compared to the two normal haplotypes. And so what we can infer from this is that one of the events that occurred at this locus was a complete loss of that normal haplotype 1. The next thing we can see here is in the remaining haplotype, there is a 12 kb deletion, which spans at least one exon there. And then lastly, if we look upstream at the promoter region, we can see the methylation pattern. So here red is hypermethylation and blue is hypomethylation. And what we see here is tumor-specific hypermethylation of that P10 promoter in the remaining haplotype. So this would lead to downregulation of P10 gene expression. And then what that means is that we have a total loss of function of this critical tumor suppressor gene in this cell line. And the only way we were able to work out what happened was because of HiFi's capabilities. For whole genome sequencing, another really cool thing that we've implemented recently with the Spark Revio chemistry is the ability to call M6A. And so this lets us look at another layer of epigenetic regulation through the FiberSeq assay to measure chromatin accessibility in a long read context. And so the way that this works is you take cells and you use a methyltransferase to artificially label the accessible chromatin regions with dense 6MA marks. And that's the little purple asterisks here. And then you extract the DNA and sequence it, and you can read out those M6A marks to identify the regions of open chromatin. So in a single whole genome assay, you get the genetic variants, the 5MC methylation status, as well as the chromatin accessibility, which lets you look for new types of biomarkers related to gene regulation in cancer. And as an example of this, the group who invented FiberSeq recently applied this method to an individual from the Undiagnosed Diseases Network who displayed a bunch of different phenotypes, one of which was bilateral retinoblastomas. And they found a balanced translocation between chromosome 13 and chromosome X, which resulted in inappropriate X inactivation of the derived chromosome here, which contains the RB1 locus. And so these cells have a first hit inactivation of RB1 through the mechanism of chromatin accessibility and X chromosome inactivation. Turning now to RNA, which is where Dr. Salomonis will focus his talk, the advantage of HiFi in cancer transcriptomics is really the ability to characterize the full spectrum of RNA dysregulation in cancer. So that's what we're showing here. While we know cancer is a disease of the genome, much of the effects of these genetic mutations are actually expressed at the level of the RNA. And the vast majority of cancer transcriptome studies to date have looked solely at gene expression, whether this is by expression arrays, qPCR, short-read RNA-seq. But we know that there are a lot of other ways that RNA can be dysregulated in cancer, including differential isoform usage through alternative splicing, fusion transcripts resulting from structural variants in the genome, as well as somatic mutations that actually get expressed at the level of RNA. To give you a sense of why short reads are so bad at capturing these other types of variation, here on the left, we're showing an example gene. So the DNA with exons is in pink here. And then below it are the 4 mRNA isoforms that are formed. And with short reads, which we're showing below here, you only get information about which exons are in a transcript if you happen to have a read that spans a splice junction. So that's what we're showing with these dashed lines here. However, this only tells you about a single junction. It doesn't tell you anything about the other junctions included in that transcript or the overall isoform repertoire in the assay. With long read sequencing here on the right, you literally just read off the entire transcript in one go. So you know exactly which exons are present in each transcript and in which order. So you get a complete view of the isoform repertoire without having to do any assembly. And with short-read transcriptomics, estimates suggest that it's only able to reconstruct about 20 to 40% of the cancer transcriptome. And so we actually expect that the importance of these other types of RNA dysregulation, such as alternative splicing, have actually been underestimated thus far just because short reads are so bad at capturing it. So we think there is a ton of low-hanging fruit here to be discovered just by applying long-read RNA sequencing. The last thing to note here is that with single-cell RNA sequencing in short reads, the problem is even worse because typically with short-read RNA sequencing in single cells, you're only sequencing the 3' or sometimes 5' end of the gene. So the only information you have is what is in this pink box here, as opposed to the complete isoform structures that you get with long reads. And then lastly, before I turn it over to Dr. Salomonis, I wanted to touch briefly on our Kinnex method, which was previously known as mis-seq. And this is what we and Dr. Salomonis have used to dramatically increase the throughput of our long-read RNA sequencing. So the way that this works is you start with multiple cDNAs, either from bulk or coming off of the 10x instrument, or any single-cell RNA sequencing method. We then concatenate multiple cDNAs into a single long molecule because your average transcript is only 1 to 2 kb, but we can sequence up to 25 kb. So you sequence the concatenated molecule in a single read, and then right there in the SmartLink analysis software, we computationally separate the individual cDNAs and give you back the quantitative isoform information, which you can then use to do short read independent cell clustering like you see here, because this method is increasing the throughput by up to 16-fold compared to sequencing just one cDNA at a time. So with that, I will turn it over to Dr. Salomonis to tell how he has been applying hi-fi sequencing to understand mis-splicing across different blood cancers. Thanks so much.
Great, thank you so much. And thanks everybody for the invitation to speak here today. Again, my name is Nathan Salomonis. I'm an investigator here at Cincinnati Children's Hospital in the Division of Biomedical Informatics. So a major focus of my research laboratory is to understand the role of mRNA processing in diseases such as cancer. And as a little bit of background for folks who may not remember much about alternative mRNA processing or alternative splicing, you have about 20,000 protein coding genes within the genome. Among those, about 90% or more undergo alternative mRNA processing. And what this is where you have multiple exons within a gene. And after those exons are transcribed into primary mRNA, they undergo alternative mRNA processing, including alternative splicing, which can include or exclude different combinations of introns or exons within those transcripts or affect different splice sites, resulting in different mRNA products. Those different mRNA products can give rise to different proteins that have different functional properties. These can include, for example, having isoforms that can promote cancer proliferation, ones that promote or repress certain metabolic pathways, or those that impact immune evasion. And for this reason, we're very interested in understanding what the role of these different isoforms are and if they represent novel targets for new therapies. In particular, we're interested in a class of malignancies called hematological malignancies. These include acute myeloid leukemias and myeloid dysplastic syndromes. The reason we're particularly interested in these is they tend to have a high frequency of mutations within factors that mediate alternative splicing. In particular, core splicing factors like SRSF2, SF3B1, U2AF1, and ZRSR2. And in fact, in diseases such as MDS, over 50% of the patients have mutations within splicing factors. However, what is interesting is that pediatric patients do not have mutations in splicing factors. And this is very interesting since splicing factors mutations are so predominant in a number of these hematologic malignancies. Why don't these pediatric patients have these mutations? And is alternative splicing, does it play a role in these different malignancies in the absence of these mutations? And so that's a major question of what spawned the research project that I'm going to be presenting today. And as a first step, a former graduate student in my lab, Meenakshi Venkata-Subramanian, looked at well-defined subtypes of acute myeloid leukemia, and here she's looking at a cohort called Lucigene. And each column here represents a different bone marrow biopsy from an individual patient, and each row represents a different splicing event. And so here she's running a supervised analysis to identify splicing event biomarkers for each one of these mutant groups. And what she finds is that alternative splicing is very robust at enabling reclassification of new patients that it has not seen before based on these splicing signatures. And that's compared to gene expression, where you actually often have very poor performance, in particular for splicing factor mutations and for some oncofusions in particular. And so what we observe from this is splicing events associated with different cancer lesions can enable classification and are robust biomarkers, representing robust biological programs. However, what was surprising to us is if now we perform an unsupervised analysis of alternative splicing in the same cohort, we are unable to recapitulate these same subtypes. And this was a little bit disconcerting to us on one side. And on the other side, it was intriguing because we were wondering, what are these predominant splicing signatures that we observe? And we could not attribute these to batch effects or other technical variables. So this appeared to be a robust biological signal. So to address this interesting question and this challenge, we set out to determine whether genetics and de novo subtypes of cancers could be identified in an entirely unsupervised manner from splicing data alone. Can we computationally identify likely causal regulators, such as particular splicing factors, that give rise to distinct splicing signatures that are observed? And are there novel broad splicing subtypes, like I showed you in the last slide, that actually are associated with prognosis, and more importantly, could be potentially observed across cancers? So to address this, we developed a multifaceted computational workflow called OncoSplice. And OncoSplice has 4 components to it. The first is called multipath PSI. This was a robust algorithm to quantify splicing events, both known and novel. These include, for example, cassette splicing, intron retention, alternative promoters, alternative splice sites, but do not assess, for example, alternative polyadenylation, because these analyses are focused on exon-exon and exon-intron junctions. We've since benchmarked this approach relative to a number of other algorithms, and it is able to perform quite robustly, often outperforming those other approaches. And then importantly, we use the splicing events that are quantified from the cancer patients to identify subtypes. And this includes both broad, highly shared subtypes and minor subtypes such as splicing factor mutations. And each one of these needs to have robust splicing signatures associated with them to be identified, as I'll mention a little bit more in the next slide. Our analysis also included a workflow called Bridger that supervises the identification of splicing-defined subtypes based either on known mutations, correlation to splicing factor knockdown signatures, or the correlation of RNA binding protein expression to splicing. And finally, in collaboration with Matt Weirich's group here at Cincinnati Children's, we developed a workflow called RBP Finder based on their RELI workflow, which takes ClipSeq, genome binding or transcript binding profiles, RNA binding protein expression levels, splicing factor motifs from the CISBP2 database or CISBP RNA database, and splicing event information to infer what the causal splicing regulators are for splicing signatures. And so this is available online in GitHub. The core of this approach is an algorithm called Splice-ICGS. This is an iterative workflow that first identifies these broad splicing-defined subtypes using a type of unsupervised clustering called HOPAC clustering in R. After this, it performs sparse NMF to define discrete subtypes. And from the discrete subtypes, It then performs a linear SVM step to reclassify the samples into discrete patient populations as clusters. And then it actually goes back and it actually removes the splicing events that are associated with these signatures, repeats this process to find finer and finer subtypes. We were able to benchmark this approach against a number of fuzzy and biclustering methods, and Splice ICGS is nearly able to identify all prior defined, genetically defined subtypes, whereas alternative approaches were not able to do so. So we're quite confident this approach was both sensitive to identifying known subtypes and identifying novel subtypes. For the purpose of discovery, we focused on 4 AML cohorts: Lucidgene, TCGA, BDML, and a pediatric cohort called Target, either sampling from bone marrow or blood. And this included samples spanning over 1,000 different patients. Because some of these cohorts did not have highly defined mutations or gene fusions, we used an integrative approach to identify genomic variants from the RNA sequencing data, including GATK for RNA, fusion catcher, a workflow called KM and Mystique, and use the associated splicing quantification data to then further perform these unsupervised analyses or supervised analysis of alternative splicing, such as shown here, and infer functional impacts, for example, nonsense mediated decay, based on the predicted mRNA sequences. And so these are the results of the analysis when we focus on Lucidgene. And first, we identified 9 prior defined subtypes. These were genetically defined subtypes of AML, and this was performed in an entirely unsupervised manner. One of the intriguing observations from this analysis was a subtype that we annotated as HNRMPK, knockdown variants. And the reason why this was intriguing is this is not defined by a single mutation, but by a number of loss of function mutations, copy number variations, and even intron inclusion that resulted in non-functional HNRMPK protein. And as such, this would not have been really readily identified by genetic-based workflows to identify this splicing-defined subtype, which is a robust splicing, produced a robust splicing signature. In addition, we identify a number of previously undefined subtypes. We've named these based on the enrichment of different mutations or oncofusions where we had that data available. In other cases where we didn't have enrichments, we just listed these as the identified splicing signatures with a number. And in some cases, we have data that's correlating the splicing signature to specific putative regulators that are inferred. Interestingly, however, the 2 most broad subtypes, which each constitute about 40% of the patients, were associated with broad splicing dysregulation that we attributed to dysregulation of specific splicing factors. And this is because when we view the predominant splicing signatures associated with mutations in U2AF1, in particular S34, or splicing events that are associated with this mutation in this gene SRSF2, this U2AF1-like and SRSF2-like splicing signature appear to phenocopy broadly the splicing that's observed in these genetically defined subtypes. And these are gain-of-function mutations. So this is not a loss-of-function associated splicing signature. And this is quite interesting because these are fairly rare mutations within the context of leukemia. But here observed here, with these really broad splicing patterns that really oppose each other, which we found fascinating since it's not really been previously described. And in fact, we look at the number of splicing events that are induced with all of these different known and novel subtypes of splicing, and we compare that to the number of differentially expressed genes, we find that not only are these 2 novel splicing subtypes, SRSF2-like and U2F1-like, defined predominantly by alternative splicing, but have over 7,000 or 6,000 splicing events each, unique splicing events identified each, making them, far and away the most broad and impactful events that we observe within all of leukemias. If we look at the types of splicing events that are induced by these splicing signatures, they're nothing to write home about in particular. And predominantly, they're results mostly in exon inclusion, not in intron retention, as opposed to other splicing factor mutations such as SF3B1, results predominantly in alternative 3' splice sites, or ZRSR2, which results predominantly in intron retention, which have been previously described in the literature. Another striking and intriguing observation is that the splicing of transcripts do not appear to be random, but instead result in the coordinated increase in exon inclusion, specifically in these U2AF1-like patients, versus exon exclusion or exon skipping, which results in much longer isoforms. For example, here I'm showing this gene caspase-9, which results in the inclusion of exon 7 through exon 10, whereas SRSF2 is skipping, by and large, these different exons, resulting in a truncated, much shorter protein. And in fact, if we now apply an algorithm in our software alt-analyze that infers what are the protein and domain impacts within each one of these conditions, where we compare all splicing events to all other patients, we find that U2AF1 and SRSF2 are on opposite sides of the spectrum, with U2AF1-like patients resulting in the least impact to protein isoforms, resulting in the longest protein isoforms, whereas SRSF2-like results in some of the shortest protein isoforms with the most strongest domain impacts, resulting heavily in nonsense-mediated decay, which we've never observed this type of phenomenon before when looking across different conditions. So again, this is something biologically unique and coordinated. And in fact, if we now look in TCGA, where we have survival data, we do see that the U2AF1-like splicing subtype is associated with poor prognosis. This is not something we would have initially predicted, since you would expect that potentially maybe truncated isoforms would result in poor prognosis, but that's not the case. In fact, longer isoforms appear to be resulting in poorer prognosis, and SRSF2-like is resulting in better prognosis. And in fact, now if we look in pediatric AML, where we also see these U2F1-like and SRSF2-like signatures, I didn't mention this before, but we observed this in all AML cohorts, this U2F1-like and SRSF2-like splicing signature. It again is associated with poor prognosis. And I'd like to reiterate that there are no splicing factor mutations within these pediatric patients. So this is not a splicing factor mutation-driven event. This occurs through some independent mechanism. So there are a number of questions that arise from these preliminary analyses. And so what we wanted to do is really understand what is regulating U2F1 and SRSF2-like splicing in AML. Possible regulators could be defined through a variety of different mechanisms. And so it could just be that there really is cell type heterogeneity within the bone marrow sample, and that there's, just a high amount of certain progenitors or a certain type of leukemic stem cell, and that heterogeneity is really driving these differential splicing, and that's why we see this effect. In fact, that hypothesis has been previously proposed in the literature. It could be that you have differential splicing factor expression or activity, potentially even hopefully due to, in principle, to U2F1 or SRSF2. And we potentially even could have non-classical modes of gene regulation, including, for example, circadian rhythm. And so these are something we want to investigate. We also want to understand whether there is a therapeutic utility to focusing on these different splicing signatures. So, for example, is U2F1-like splicing stable over time? And are there even potential inhibitors of them, this splicing, that we could consider for clinical applications? And so to first address the question of possible regulators, in particular, if there's single cell heterogeneity, this is what we focused on. And we could have focused on this using deconvolution approaches, single cell deconvolution approaches, which is something we did try and can provide some information. But ultimately, really the best way to address this problem without having to focus on very biased approaches like bulk RNA sequencing, even sorted populations, was to perform an entirely unsupervised approach. And for this, we turned to Kinnex sequence, Kinnex long single cell long read analysis. And so fortunately, we actually already had bone marrow biopsies from different patients in which the leukemic stem cells associated with the AMLs were knowable. And the reason these were knowable is these patients, which were previously described in this paper from Craig Jordan's group, who shared these bone marrow samples with us, we were able to obtain this bone marrow, which had already xenografts, PDX models generated from them, to determine whether it's more of a primitive leukemic stem cell or a more mixed primitive or monocytic leukemic stem cell. The more monocytic leukemic stem cells tend to be poor prognosis and treatment resistant. And each one of these bone marrow biopsies was subjected to 10x chromium analysis to derive single cell libraries in which short read site-seq was performed on this with deep sequencing to derive these 150 nucleotide 3' end fragments and antibody-derived tags on 132 different antibodies. Using these 10x libraries, which we previously generated and published, we're now able to take the cDNA and through a collaboration with PacBio, generate a full-length long-read sequencing using this Revio platform and the Kinnex single-cell long-read solution. This produced about 50 to 100 million reads to begin with, which generated about 3,000 to 5,000 UMIs per cell, and on average about 1 kb long sequences, although in many cases we had much longer full-length RNAs that were produced that we can focus on. And a first question is, how concordant is the gene expression that we would get from this platform relative to standard Illumina-based sequencing? And in fact, since we do have the exact same cells profiled with Illumina and with Kinnex, we can actually perform head-to-head analyses. Here we focused on a single cell population. This is a population called MPP1 cells. And here we can see that there's a high degree of concordance between the Kinnex and the Illumina-based analyses with lower expected sequencing depth using Kinnex. And in fact, we're also able to call known genetic lesions within these samples, including specific mutations in each one of these 3 patients, deletions, and frame shifts. And so this was quite robust for different variants. For example, in the case of NPM1, we have over 14,000 reads in this sample with this previously exome-defined frame shift. So the majority of cells profiled will have this variant, whereas in others we had fewer reads, only a few hundred, which is associated with the detection of these transcripts based on the expression of the gene and the position of this variant within the transcript, whether it's closer or further away from the 3' end. However, we really needed a robust solution to perform a number of unsupervised analyses and supervised analyses that were focused on alternative splicing. We were not able to find those tools that really existed to do this, and so we ended up creating our own. And this is this workflow I'm showing you here, which we're calling alt-analyze LR, alt-analyze long read, is a part of our Python 3 version 3 of our workflow called alt-analyze 3. Which combines both tools for bulk RNA seq analysis. So here you can take BAM files and perform standard quantification of exon-exon junctions, exon-intron junctions to produce these bed files and H5ADs from these sparse annotation files using this ensemble gene model and isoform model. And we can couple that with a long-read sequencing data to perform automated splicing quantification and long-read quantification analyses, including differential percent spliced in, PSI analyses, differential isoform abundance, and different isoform ratios, comparing individual cell states to each other or between different biological conditions. And we're building this out into a larger framework that looks at splicing neoantigens and performs other unsupervised analyses, such as OncoSplice, downstream of these results. And one of the key components of this alt-analyze long-read workflow is the integration of different samples at the level of transcript models and transcript expression. And so this begins with a gene model. Here we're using Ensemble's gene model. We collapse all the known isoforms that we can assemble from them into this annotated gene model where each one of these exons are called out as a different exon number and associated intron numbers. We then take these ensemble models and we combine them with the isoform models that are obtained from each individual sample here, indicated by sample 1, sample 2, or sample 3 here. And each one of these are different isoform IDs. We can collapse these different isoforms into groups where they have overlapping exon-exon junctions or splice sites or polyadenylation sites. And from the collapsing, then we can identify unique isoforms that then serve as references that we can basically then feed back to the individual samples. So now this is our consolidated reference. So all the different samples now will have, even though you have an isoform that's observed initially in sample 3, you'll be quantifying it in samples 1, 2, 3 as well. And we take this isoform data, we're able to assign the different cell barcodes to different clusters. This can be using tools within AltAnalyze 3 or publicly available tools, such as we have an azimuth browser based analysis tool that allows you to upload your single cell data to it and assign the cell annotations to well-defined single cell populations from previous studies, ours or others. And then you can take these single cell annotations and perform cell state level comparisons, either one cell state against another or different conditions against another, where you're quantifying both the abundance of different isoforms within these different cell states, or you're quantifying the abundance of individual splice junctions, intron retention events, or alternative polyadenylation, or alternative promoter events to each other. So from these different statistical analyses, we get differential isoforms, differential splicing events, and we even get isoform impacts that are derived through in silico translation of those quantified isoforms. I should note here that the collapsed isoform references we're using are the longest isoforms. So even if you have shorter isoforms that are detected due to technical issues, the analytical workflow is actually inferring what the longest ref consensus isoform will be and assigning that to the other associated transcript reads in each individual data set. I mentioned before we're using a standard reference here. This is a bone marrow atlas, which is a fairly comprehensive atlas, which was produced on 3 different flow-based captures in 8 different patients. Spanning over 500,000 cells from healthy bone marrow in 87 discrete cell states. And we can use this now to transfer these labels into the leukemias in which we're going to see different biases in cell populations that are not normally seen or that are potentially normally seen in normal hematopoiesis but are enriched in the leukemia. And so initially, what we're looking at here is our 3 patient samples. Sorry, they're colored here on the next slide. But we see that there's a high degree of variation in some cell populations. So I'll just skip to this one. In AML7, for example, you have a low percentage of cells that are derived from this intermediate monopopulation. However, in AML12, the majority of cells identified are from this intermediate population. And so you see these different cell frequencies from the mass-seq that are identified. And one of the key questions we needed to identify here is, was this, do we see different splicing patterns associated with different cell types, or is U2AF1 and SRSF2 splicing conserved across different cell population? And it actually is the latter, which is suggesting that the splicing we observe with U2AF1 versus SRSF2 is not cell type specific, but rather it's intrinsic. It's patient intrinsic. So in each one of the cell populations in these green triangles, the cell states from this one patient, this AML7, are all U2AF1-like, versus from AML12, they're all SRSF2-like. So there is an intrinsic splicing program that differs within these AMLs associated with U2AF1 or SRSF2-like splicing, this resulting in longer or shorter transcripts. And so to really see how well this is performing, we can look at splicing events that we previously identified from our bulk RNA-seq. So here we're looking at patients with U2F1S34 mutations, U2F1-like, SRSF2-like, or patients with these SRSF2P95 mutations. And here, for example, we're looking at inclusion of this exon here, exon 8, in UPP1, which is really spliced out or skipped in SRSF2-like. And now we can look at the full-length isoforms with the long-read single-cell Kinnex, and we can see here the isoforms with inclusion of this exon based on IGV visualization or skipping, and we see different ratios here in U2F1-like versus SRSF2-like that match what we see in our bulk RNA-seq analyses. Similar, here's this target gene IRAK4, which I'm going to be talking about a little bit later, in which its exon 4 is skipped in the SRSF2-like and SRSF2 mutations, and we see a switch from this longer isoform that has exon 4 being the predominant isoform in U2F1-like to it being the rare isoform in SRSF2-like. But in addition to this, we identify a number of novel exons which have different isoform linkages to each other that are not been well described. And since IRAK4 is a therapeutic target in AML, this represents an interesting opportunity to understand the function of these different isoforms and their contribution to leukemic cell growth in the future. But importantly, we can now actually quantify alternative splicing at a higher resolution than we've done in the past. And in particular, we can annotate these, for example, intron retention, much more accurately from the long-read data. And so when we look in just one of those cell populations in the AML monocyte progenitors, and we look at splicing events that result in increased inclusion or proximal splice site selection, we tend to find increased in U2F1-like versus SRSF2-like. We observe increased cassette exon inclusion and decreased intron retention, whereas in opposition to that, if we look at exon exclusion, we see decreased cassette exon skipping and increased intron detention, or actually moving away from intron retention is the way that you think about that. But we also observe a large number of alternative polyadenylation events that are predicted, which represent over half of the events identified. And now we can actually view with the Kinnex data these specific alternative polyadenylation sites here within the data, including those that result in longer 3' UTRs, for example, in U2F1-like versus the SRSF2-like patients. We can use this same data to actually now prove effectively that U2AF1 is resulting in much longer protein isoforms because we have the full-length isoform sequencing data versus SRSF2 and look at their specific protein products. So that really does address the question of whether it's an intrinsic program, of which it is, versus stem or progenitor cell variation. But that doesn't address really what the regulatory splicing factors are here, and whether those are due to different splicing factor activities or different expression of these different splicing factors. And so we looked at splicing factor activity first, and the way that we can do that is we can take, we can look at these splicing events, see where the different splicing events occur at known U2AF1 splice sites, and in U2AF1 there's the canonical splice sites shown down here, the splicing signature, and the non-canonical splice sites motif patterns shown with S34. And indeed, what we observe is that the majority of U2AF1-like splicing events do not follow the mutant splicing profile. They follow the canonical profile, but because you are hyperactivating splicing here, here you have U2AF1-like splicing, you're resulting just based on the background of how many more splicing events you see throughout the genome. We see tens of thousands of splicing events in some cases. You see that there's a byproduct of these S34 specific targets being selected for. So we think this is due to promiscuous activation of splicing, so U2F1 just binding broadly throughout the genome, as opposed to it binding more specifically to these S34 targets. when we look at expression of different splicing factors and their correlation to either U2AF1 or SRSF2-like splicing, we find that several splicing factors are nominated as putative regulators of these splicing profiles. In particular, WDR77 and PRMT5, which form a complex to regulate splicing of small nuclear proteins within the nucleus, in which, importantly, there have been drugs developed for PRMT5 to inhibit its activity. And we can confirm the activity of these splicing factors now through, for example, knockdown. So if we look at U2AF1 siRNA, we see that it actually rescues thousands of splicing events relative to those observed in patients here. We see the opposing splicing signature induced with knockdown of U2F1. We see the same thing with WDR77 knockdown. And if now we treat the AML or MDS cells with this inhibitor of PRMT5, we actually again can see this correction of splicing events that are induced within the patients, suggesting both the splicing factor complexes are involved in U2AF1 splicing regulation and the potential therapeutic control of splicing using PRMT5 inhibitors. And then finally, we want to look at non-classical modes of splicing regulation, particularly circadian. And this is based on other observations that we've had in our lab, that splicing is frequently concordant with the time point of day in which the sample was collected from using tools like Cyclops. And in fact, if we look at healthy bone marrow cells from normal donors, we see the core U2AF1 and SRSF2-like splicing signatures viewed even in the absence of leukemia. And this suggests, again, that this is an intrinsic phenomena associated with hematopoietic stem cells that may have something to do with the time of day they're profiled. And in fact, if we look at these healthy donor induced genes or repressed genes versus those observed in AMLs, again, between U2F1 and SRSF2, we see this enrichment in factors associated with circadian regulation, including timeless, a CRYZ, PER2, CRY2, and PER1. And indeed, if we now actually take our bulk RNA-seq splicing data and we use these temporal ordering approaches, particularly Cyclops, to order these in particular circadian phases, we see that there's a strong concordance with the observation of U2AF1-like patients and SRSF2-like patients with these splicing profiles, suggesting, again, this may be linked to circadian dysregulated gene program. So finally, I'd like to focus on what is the potential therapeutic utility of this. And if there is a therapeutic utility, these splicing signatures need to be stable over time. And indeed, that is the case. So if we look at diagnosis and relapse in this independent cohort that's been previously described from Cornell, we see there's a high concordance between diagnosis and relapse. And again, if we look at beat AML at similar associations looking at multiple time points of therapy, we see a strong degree of concordance here between U2F1 and SRSF2 splicing over time. So these are largely stable splicing signatures. As I mentioned before, I showed you that we tested a PRMT5 inhibitor on these cells, and it was able to reverse the splicing signatures associated with U2AF1-like splicing. And now, if we look at different days of treatment with PRMT5 in this MDS AML cell line, MDS-L, we see decreased viability of these cells over days in culture. We see decreased NF-kappa B expression, and we see an increased myeloid differentiation phenotype associated with PRMT5 treatment, quantified either through CD14, CD11B expression, looking at the histology of the cells, and by flow cytometry. And one of the targets, the main targets that were predicted from PRMT5 modulation was IRAK4. Again, with inhibitors exist for IRAK4 as well. But if you inhibit with PRMT5, you see loss of inclusion of the U2AF1 associated exon 4. This is also reflected at the protein level in terms of this longer isoform IRAK4L with inclusion of exon 4, and a decrease in protein expression over time with PRMT5 treatment. And with IRAK4 knockout, we also see this increased cell survival phenotype, which is quite promising in both THP1 cells and MDSL cells with IRAK4 expression, suggesting that this is somewhat recovering or rescuing the AML phenotype, at least in culture and in associated xenograft models, as shown here. And so in summary, alternative splicing defines novel poor prognosis cancer subtypes. Long-read sequencing analysis confirms that U2F1-like splicing is tumor cell intrinsic, resulting in longer protein coding transcripts, and that U2F1-like splicing results from an overactivation of U2AF1 activity as opposed to phenocopying the activity of the mutant U2F1 protein. And finally, U2F1 splicing and AML viability can be reversed directly either through PRMT5 inhibition or through knockout of its target IRAK4. And so with that, I'd like to thank our many collaborators who have helped with these studies outside of members of the Salomonas Lab, including members of the Lee Grimes Laboratory, the Dan Starzanowski Laboratory, Matt Weirich's group, all at CCHMC, Craig Jordan's group at the University of Colorado, Denver, PacBio, who actually performed some of the single cell long read sample preparation and sequencing for us. Members of Amit Verma's group out at Albert Einstein College of Medicine, who drove the PRMT5 inhibition studies, and their collaborators at Prelude Therapeutics, as well as funding from National Cancer Institute, an Evans MDS grant, Cincinnati Children's Research Foundation, and the University of Cincinnati. And with that, I am happy to answer any questions you might have. Thank you.
Awesome. Thanks so much. And thanks so much, everybody, for sticking on and listening. In the last couple of minutes here, we just have a couple of questions that we wanted to go over. And so please, if anybody else has any, please feel free to submit those. So maybe a question for Nathan here on the technology used. So OncoSplice only looks at splice sites. Is it able to look at alternative first exons or poly A tails?
Yeah, so it's a good question. In principle, it can analyze any type of mRNA processing regulatory data you throw at it. The way that this version is configured was with a tool called AltAnalyze 2. This was a bulk RNA-seq splicing quantification workflow for Python 2.7 or Python 2. We've been upgrading our workflows to work in Python 3. Python 2 is legacy. It's not really supported anymore. We have Docker containers for it, but it's still not ideal. So we've been updating all of our workflows to Python 3 and refactoring and redeveloping a lot of these workflows. So OncoSplice 3 actually work in addition to exon-exon junctions, which include alternative promoter regulatory as first exons, will include alternative 3' splice, alternative 3' mRNA processing, alternative polyadenylation. So, in particular with the long read analyses, as I showed, you can perform those alternative polyadenylations analyses with that now.
Awesome, thank you. So another question here is sort of more generally, we're talking about mis-splicing. So how do you define mis-splicing? Is that, would you say the same as promiscuous splicing, or is there a different definition?
So this, yeah, this is a weird concept. And so for folks familiar with MDS, and in particular these splicing factor mutations, these mutations engender the new protein to bind to different consensus splice sites. So it has a different RNA recognition properties. And so U2AF1, for example, in terms of the S34 mutation, has a slightly different binding logic than the canonical U2AF1 protein. What we find is, but that does not mean that the canonical protein never binds to the same splice sites that U2AF1 mutant does. It just does it with less affinity. And what we see here is that in AML, without these mutations, if you super ramp up the activity of alternative splicing in U2AF1, like you have a byproduct, a bystander effect, that these same U2AF1 S34 splice sites just are tend to be bound because you're binding to thousands and thousands of more splice sites than you typically do. So it's a byproduct. It's not a targeted effect, it's an off-target effect.
Cool, that's helpful. That makes sense. So this is sort of a more general question. So I'll let Nathan take this and then I can chime in if needed.
Yeah.
Other than what you presented, is there specialized software for fusion transcripts as well as alternative splicing?
So for the long read fusion detection, there are specialized tools that I believe a number of groups have developed, and I believe these are available linked to from the PacBio website. But they are fairly robust for detecting, for example, oncofusions in the RNA data. And there are a number of other tools that do other cool things with long read data, single cell long read data. So for example, there's a package that I don't remember the specific name, but it's a tool to identify MHC allele sequences. So it identifies HLA genotypes from long read data much, much better than even bulk RNA-seq data. Because the links are phased, they're not phased, but you have a full-length read, you don't have to infer which specific isoforms are co-occurring together on the same isoform. So we've been using that for neoantigen-based analyses, and that's quite nice.
That's super cool. Yeah, and I think actually in one of those pieces of literature that's attached on the console, I think there's actually a app note on a bunch of different bioinformatics tools that you can use for long-read RNA sequencing. So if anybody is curious. So here's another question. Well, this was a limited number of patient samples. How much variability are you seeing patient to patient and cell to cell?
Yeah, that's right. So we only did the long-read single-cell analyses on 3 patients in this study. We've now extended it to about 5 or 8 or something in that area. We do see a lot of variability in the amount of U2AF1-like and SRSF2-like splicing, and this is because it is a gradient of splicing that we observe that has this circadian phase-like distribution. And so we see that, however, in all the cases we have looked at, you do not see that different cell types have different U2AF1-like splicing. They all have typically the same degree of U2AF1-like splicing together as a group, and they do not differ by cell type, but they differ by patient. But it is a small cohort.
So I guess maybe related to that, what are your plans to do next? What would the next steps in this study be for you?
Yeah, so I think there's a lot of cool questions here that we could, go off of. And so specific splicing events, we've nominated, like over 10,000 splicing events that have really extreme differential splicing within leukemia. Some are never found in normal healthy cells when you have these kind of pseudo U2AF1 and SRSF2-like phenotypes. And many of these are in oncogenes. And so specific follow-up validation studies would be of particular interest for us. For example, using morpholinos are some of these therapeutic targets. The best demonstrated example of this is IRAK4. So if you knock out IRAK4 or develop drugs to it, it is itself a therapeutic target and can result in recovery of leukemic drug-resistant cancers. And so there are clinical trials that are ongoing for that. And so similarly, we'd expect that many of these other genes that we've identified are interesting therapeutic targets. We also are considering some of these as potential neoantigens using that SNAP workflow I mentioned before, which was published last year. And so does this broad splicing result in novel isoforms that can potentially leverage for new cancer immunotherapies such as cancer vaccines?
That's cool. That's super interesting. I know we are at time, but there's a couple of other questions that have kind of come in the chat. So for the three individuals that you did the long read single cell RNA sequencing for, did those individuals have splicing mutations, or did they just exhibit this mis-splicing phenotype?
So one of them did have an SRSF2P95 mutation, and they are, I believe, oddly enough, I think they may be the U2AF1 like sample. And so it's not always the case that SRSF2P95 or U2AF1, S34 have the same splicing pattern as the U2F1 like or SRSF2 like, but it tends to be a little bit more frequent. We've been trying to sequence more patients that do not have splicing factor mutations. But right now, a few in our cohort do have S34 or SRSF2 mutations.
Cool. Here's another one. The sequencing here was very deep. How many reads do you feel are required to characterize splicing in these samples?
That is an excellent question, and it's an ongoing challenge for us. It's really dependent on the number of cells you have within a cell population and the resolution of your clustering. So we are using pseudo bulks here, and we're collapsing the spliced reads and isoforms at a cell population level to get sufficient depth here. And that's typically what is needed. So you effectively have these mini bulks from a single cell experiment. We've performed analyses with about 100 million reads, which is the kind of upper end that you get with the Kinnex plus Revio. And that is typically sufficient if you're doing large cohort analyses. You probably wouldn't want to sequence the crap out of individual samples. I think it's always better to sequence more samples. You may have to throw out some pseudo bulks due to low depth is the challenge there, or use a lower resolution clustering.
Cool. That makes sense. Just a few others here. I think I may have missed it, but what do you think causes the enhanced U2AF1 splicing activity in that U2AF1-like signature? So was this linked to splice factor expression, like the expression of that factor?
It's a great question again. In the case of U2AF1, we do see higher MYC expression transcriptionally, which is MYC regulates PRMT5 and WDR77 expression, both of those, which are upregulated. It gets more complicated. WDR77 itself is alternatively spliced and associated with both prognosis and this U2AF1-like splicing phenotype. But we do believe that the PRMT5 WDR77 pathway is likely the key regulator here, especially because if you knock down WDR77 or U2AF1, you have the most significant impact on reversing the splicing phenotype. But it's a good question. We don't know, the entire upstream set of putative regulators, but that's our best candidate right now.
Cool. That's really interesting. Awesome. So I think we've gotten at least to most of the questions here, and we can also follow up with anybody who didn't get their questions answered. But thank you so much, everyone, for joining us, and we hope to see you at our next webinar. Thanks again, Nathan.
Thank you.